Hello students, this is your instructor Abid John and today's lecture is about DNA sequence databases. DNA sequence databases. The Embl, the European Molecular Biology Laboratory, the nucleotide sequence database from the European Bioinformatics Institute. It includes sequences from direct author submission, genome sequencing groups, and from the scientific literature. Embl collaborators and model systems. The database is produced in collaboration with the DNA Databank of Japan and Genebank. The participating groups each collect a portion of the total sequence data reported worldwide. And all new and updated entries are then exchanged between the groups on daily basis. Embl contains millions of entries, representing more than 15,000 species, but with some model systems predominating that collectively contribute to more than 50% of the resource. These model systems include Homo sapiens, Caenorhabditis elegans, Saccharomyces cerevisiae, Mus musculus, and Arabidopsis thaliana. Genebank. Genebank, the database from the National Center for Biotechnology Information, incorporates sequences from direct author submissions and large-scale sequencing projects. To help ensure comprehensive coverage, the resource exchanges data with both Embl and DDBJ. Divisions of Genebank. The increasing size of the database, coupled with the diversity of data sources available, have made it convenient to split Genebank into smaller, discrete divisions. These are summarized in table. We have more than 20 subdivisions in the Genebank. Some of them are mentioned here. PRI, ROD, MAM, VRI, INV, PLN, BCT, VRL, PHG. This division facilitates fast, specific, and comprehensive searches by restricting queries to a particular database subset. The structure of Genebank entries. A Genbank release includes sequence files, indices created on various database fields, and information derived from the database. Originally, Genebank was made available on CD-ROM, which proved to be a convenient mechanism for widespread and relatively inexpensive distribution. However, as the size of the database grew, the number of CDs required to contain it became very difficult, both for the producers and for the users. So today, Genebank is available only via FTP, File Transfer Protocol. Each entry consists of a number of keywords, sub-keywords, relevant associated sub-keywords, and an optional feature table. Its end is indicated by a terminator. The positioning of these elements on any given line is important, and any line beginning with a blank character is considered a continuation from the previous section. Keywords description. The first keyword is locus. The locus keyword introduces a short label for the entry that may suggest the function of the sequence. This line summarizes other relevant facts, including the number of bases, source of sequence data, and division of the database, and date of submission of entry. The definition line contains a concise description of the sequence. The accession line gives the accession number, a unique constant code assigned to each entry. The NID line supplies a nucleotide identifier intended to provide a unique reference to the current version of the sequence information. The keyword line introduces a list of short phrases describing gene products and other relevant information about the entry. The source record provides information on the tissue from which the data have been derived. There is a sub-keyword within the source, and that is the organism, which illustrates the biological classification of the source organism. The next keyword is reference. The reference record indicates portion of the sequence data to which the cited literature refers, and the sub-keywords within the reference, which are authors, title, and journal, provide a structure for the citation. Another sub-keyword, the midline, is a pointer to an online medical literature information resources which allow the abstract of a given article to be viewed. The next keyword is the feature. The feature keyword introduces a section that has its own structure and whose purpose is to describe properties of the sequence in detail. Within the table, links to other databases are made. Genomic coordinates for the 5' and 3' untranslated regions for the coding sequence and the polyadenylation sequences are given in feature table. The next keyword is base count. Base count record details the frequency of occurrences of different base types in the sequence. The next keyword, the origin line, notes the location of the first base of the sequence within the genome. The nucleotide sequence itself follows and the entry is terminated by marker, this symbol. This is the bibliography. Keep calm, stay curious. Allah Hafiz.